The next part of the kidney we're going to be looking at is the loop of Henle right here. So it has the descending limb, descending going down, and then the ascending limb of the loop of Henle. Now this part of the nephron is very important because it functions to maintain a hypertonic environment inside the kidney. You'll wonder why would you want a hypertonic kidney? The reason is that you will want to reabsorb as much water as possible. You don't want to drink water and then pee it all out. You need to keep that water inside the body. Now, if the outside tissue of the kidney is hypertonic, it means as your filtrate passes through the loop, it will reabsorb water. It will keep water inside the kidney instead of peeing it out. Now, how does the loop maintain a hypertonic environment here in the medulla part of your kidney? So let's show you a diagram. So allow me to borrow this animation of the counter current multiplier system. So as you can see here, so this is your kidney, cross section of your kidney, and this is where, okay, it's zooming in right now, that's where your nephron is located. So the Bowen's capsule and the proximal convoluted tubule is part of the cortex, the outermost layer of the kidney. However, the loop, as you can see, it's inside of the kidney. This part is called the medulla. So this medulla right here has a hypertonic environment, while the cortex is not. And the reason why it's hypertonic here is because of the loop. Now, what's happening here in the loop? Let's zoom that in. All right. So you have here the descending and then the ascending limb of the loop. First of all, you need to know that the descending limb is permeable to water only. So only water can go out of the descending limb of the loop. On the ascending part of the loop, this part has sodium pumps, and this can pump sodium out of the tube. So what happens is, let's try this with some numbers. Let's see if this has a number. Hold on, please. All right. Sorry, my mistake. This one doesn't have numbers. Let me look for one that has numbers. So the osmolarity of the filtrate after it was filtered out from the proximal convoluted tubule is about 300 millimoles. Hold on. So it was all 300, and then we moved it here. The ascending part of your loop is permeable to sodium, so it's pumping sodium out. As it pumped out 100 milliosmoles, so the 300 became 200, and then the medulla here is now slightly higher osmolarity, that's 400. However, the descending limb is permeable to water, so what will happen is water will, through osmosis, go out of the loop until it reaches an isotonic solution. So let's fast forward that. There. So it's now the same osmolarity as the outside, or as the medulla, so it's now isotonic. And let's fast forward this filtrate again. So, all right, so 400 here again. The ascending limb of the loop of Henle, please remember, is capable of establishing a concentration gradient of 200, a difference of 200. So it will pump out sodium again into the medulla. So it was 400. It was all 400. And let's wait. So this is going to pump 100 out. All right, so that will make it 300. Your medulla is now 500. And obviously, the descending part of the limb, as you've seen earlier, this will also, by osmosis, water will come out to equalize the osmolarity, so it's isotonic to the medulla now. So let's move up that 500, and the same thing will happen the ascending limb will pump sodium out to establish 200 concentration gradient. So now that's 600 and 400. And water will come out to equalize that. So that's 600 now. So this is going to keep happening until we've established about 1,200 osmolarity here in the medulla. The osmolarity of the human kidney is about 1,200, and it differs from species to species. Other animals, such as the ones living in the desert, they have to store more water, so they will have a higher osmolarity here in their kidney, and at the same time they might have longer loops of Henle, so that they have more tubes and more chances to concentrate the environment here in the kidney. And this whole process is called the counter multiplier system. So counter meaning this pumps it out, this pumps at the opposite. Multiplier because as the filtrate keeps flowing through the loop, you have increasing osmolarity here in your kidney. Okay, let's go back to our concept map. So that's the function of the loop. Now we have to look at the function of the collecting duct. So that's the terminal tube, or the last part of your nephron. What you need to know here is the collecting duct. The cells in the collecting duct have receptors. The cells have receptors for ADH. Now ADH stands for antidiuretic hormone. This hormone is produced by the hypothalamus from the brain. So if the body detects that you have less of water inside, then it will produce the hormone, antidiuretic hormone, to trigger the cells of the collecting duct to reabsorb more water. So you want to keep more water inside the body. That's why if you are dehydrated, your pee tends to be more concentrated. It's darker in color. The smell is stronger. It's because the body responded to ADH, and these cells reabsorb. You know the meaning of reabsorb already. It will go back into the blood, or it will stay inside the kidney instead of you peeing it out. So that's the function here in the collecting ducts. Okay, so two conditions that you have to be familiar with is overhydration and dehydration. Dehydration, you're familiar with that. If you don't have a lot of water, you're dehydrated, or if you're drinking too much water, the condition is said to be overhydration. Please read about the consequences of dehydration and overhydration is also in your book. Now, if something is wrong with the kidney, then obviously your filtering mechanism is also going to be messed up, and you might end up with a disorder or the disease called kidney failure. And if your kidneys fail, it's fine, because we have a treatment called dialysis. You might also be familiar with dialysis, but basically in dialysis, your kidneys cannot filter your blood properly. So what you do is you insert a tube in your vein, then blood will come out. It will be filtered by a machine, and then the cleaned blood will be reinserted back here in your body. So that's called dialysis. And how would you know if your kidneys are failing? How do you know if you have a kidney problem? It can easily be detected by a diagnostic method called urinalysis. So in urinalysis, you're going to test your urine. If you have glucose in your urine, then something is wrong here with the Bowman's capsule. Remember that glucose is reabsorbed back into the blood. So why do you have glucose in the blood? It means you're not filtering it properly. Number two, if there's blood in your urine, then maybe parts of the nephron or parts of the ureters, bladders, and the urethra are infected. You can also detect drugs in your urine. So all of these things can be detected simply by getting a sample of urine and doing an analysis on it. So urinalysis, that's why we call it urinalysis. Last, so the waste product of the excretory system for us, it's urea. But originally it was ammonia. These are waste products of breakdown of your proteins. But ammonia is very strong. It actually has a tendency, excuse me, if you mix it in water, it can be toxic to the cells. So we convert it into uric acid or urea. Now, some animals convert it to uric acid. The example here are your birds, because uric acid is less soluble in water. So birds they will not require water to metabolize the uric acid. Therefore, it's easier for them to fly because not as heavy. They don't really need water. They can concentrate their waste as uric acid. For us, we release it as urea. For some fish, they release it as directly as ammonia, because it gets dissolved in the outside environment right away. Okay, I think that's it. So structure of the nephron, spend time studying the structure of the nephron and the functions in the different parts of your nephron. Thank you.